Assalamu alaikum wa rahmatullahi wa barakatuh. This is the second lecture in course, which is from Design 1. We will discuss the physicochemical characteristics of excipients. By the end of this lecture, the points that we should learn is we should know how to define the term excipient. We should be able to explain the rationale of including excipients in pharmaceutical formulations. We should be able to list different functions of excipients and give examples of excipients from each category. We should be able to recognize the contents of excipient monographs in compendia and in other references. Describe tests used for physical and chemical characterization of excipients. Describe the impact of changes in excipient characteristics on the formulation performance. And list the conditions that must be met by an excipient to be accepted by regulatory bodies for use in pharmaceutical formulations. So first, we should know about the dosage form design. When we want to make a dosage form or a formulation, what are the ingredients that should be included? So the main ingredient here is the drug substance, what we call active pharmaceutical ingredient. So usually the pharmaceutical dosage form is mainly intended to deliver the active pharmaceutical ingredient into certain body tissue or body organ. Okay? And is usually for local or systemic effects, so supposed to exert the effect at the site of application or should be absorbed, go to systemic circulation, and exert effect in the body tissues distant from the site of application. Okay? So usually dosage form or the formulation doesn't only contain drug substances but contains also in combination with this API in combination with one or more non-medicinal agents that we call them excipients. So excipients are non-medicinal agents, so they are not intended to produce pharmacological effect, but they serve varied and specialized pharmaceutical functions. We'll explain later on in the coming slides. So they have specialized pharmaceutical functions. So it's not a pharmacological function, but pharmaceutical. They have to serve some function that in the formulation itself. Not all the time, but most of the time in the dosage form, the total amount of excipients frequently used in greater than the amount of active drug substance. Okay, so sometimes you buy a medicine, for example, cream or ointment, and then you find that it is written on the label that concentration of the active pharmaceutical ingredient is about 2% or 1%. So this means that the other 99 or 98% is medicinal agents. They are not the API. So we call them excipients. So sometimes the excipient percentage is as large as 99% or 98% as in creams. And then sometimes you buy a syrup and then you find that it is written that the dose, the content of 5 milliliters of that syrup is about, say, 100 or 200 milligrams of the API. For example, you buy Adol syrup, the active ingredient paracetamol may be something like 100 or 200 milligrams. In each 5 ml, 5 ml usually weighs about 5 grams. 5 grams contains only 200 milligram of active ingredient or 250 milligram of active ingredient. So the rest, 4.75 milligram, is excipients. Okay? So usually or frequently, the amount of excipients in the dosage form or in the formulation is much more compared to the amount of the active drug substance or what we call API. Okay? When we design the dosage form, usually we should have a proper consideration of the physical, chemical, and biological characteristics of the APIs and excipients used in the formulation, as well as their compatibility with one another. And this is essential to produce a stable and effective drug product. Okay? When you talk about physical, chemical, and biological characteristics of API and excipients, for example, biological characteristics, you want to produce sterile or non-sterile dosage form. So if you produce a sterile dosage form like injections, then your excipients that you will use in the formulation and the API as well should be sterile. Okay? Non-sterile, then there should be microbial test and they should conform to the test. It should not exceed the accepted microbial limit. Okay? Physical characteristics like particle size, melting points, okay, many solubility and other issues, they should be also suitable for the dosage form that we want to make. They should be compatible with each other, means that they will not react and change the properties of the formulation or the API. So they can keep the drug stable during the shelf life, so the drug product will be effective. Okay? So what is the reason for inclusion of drugs in dosage form? Why you don't just give the pure API to the patient and that's enough? He can administer the pure API. This is in most cases impossible. Okay? For safe and convenient delivery of accurate dosage of drugs, especially potent ones, which are administered in milligrams or less than milligram doses, we need to include the drugs in dosage form. Safe and convenient. How can the patient weigh accurately milligrams? This is impossible. So they may take, especially for such potent drugs, may take larger than the prescribed dose, and this will not be safe. It may cause toxicity. Second thing, for convenient delivery, we want some easy dosage form that the patient can easily take, can easily administer. So, for example, vaginal delivery. How can you give a powder to the patient and advise her to administer this powder through the vagina? It will be very difficult, but it will be more convenient for the patient if you design it in the form of a pessary the patient can insert into the vagina or for example, for rectal delivery. It's very easy for the patient to take one tablet and swallow it, and 3 milligrams or even half gram of powder, and then I don't know how the patient will swallow. And you can add also difficulties in taste and other issues. So by designing a suitable dosage form, then it will be for safe and convenient delivery of accurate dosage of drugs. Okay? Some drugs also may have some pharmaceutical functions. They may protect the drug substance from destructive influences of, for example, atmospheric oxygen or humidity. So we can design a coated tablet that have a coating material that will prevent the passage of humidity, for example. Or we can make sealed ampoules, for example, that because it is sealed, then it will not allow atmospheric oxygen to pass into solution inside the ampoule. Can protect also drugs from gastric juices. Some drugs may break down, may be destroyed once they reach the acidic environment of the stomach. So you can design some enteric coated tablets. Since the coat around the tablet is made from a material that will not dissolve in acidic medium. So the tablet will not dissolve once reached to the stomach. The drug is inside the tablet and protected from gastric juices. This coat is not dissolved and protecting the inside tablet from the acidic pH. But once it will reach to the intestine, the pH will be neutral to alkaline, then the coat will dissolve, the tablet will disintegrate, and the drug will be dissolved and then can be absorbed by the GIT. Okay? Some dosage forms are designed to mask an unpleasant taste or odor. Capsules, for example, when you swallow a capsule, the part that will comes into contact with your mouth, the part that you will taste is the capsule shell. But whatever inside the capsule, whatever the taste, the bitter taste, or however the smell of it, then you will not feel it. Okay? You can also design coated tablets. Tablets may be coated with a sugar coat. Once you swallow the tablet, you will only taste the sugar coat of the tablet. You will taste a sweet taste. But inside the tablet may be a very bitter drug that it will not be pleasant to swallow as it is without the coat. You can make also flavored syrups. Most of you, I think, in your life, when you are children, you administered syrups with different flavors, probably orange flavor, blackberry flavor, and so on. So you drink a drug, medicine, but it tastes like juice. So this property is imparted by excipient. We can add flavors, we can add sweetening agents like sugar or synthetic sweeteners, okay, and they improve the taste. Also, by adding specific excipients, we can make a dosage form suitable for application into a specific site. For example, we can make pessaries or suppositories that can be suitable for insertion into body spaces or compartments, like rectum, like vagina, and so on. So we have rectal, vagina, and vagina suppositories. We can make aerosols that can be sprayed and inhaled into the lungs. We can make solutions for injection, then can be injected into either muscles or bloodstream or under the skin, subcutaneous. We can make liquid or solid dosage forms for oral administration, liquid dosage forms like syrups, solid dosage forms like tablets and capsules. We can make liquid or semi-solid dosage forms for topical administration, like lotions, it's a liquid dosage form. Creams and ointments are semi-solid dosage forms. We can make transdermal patches that can be applied to the skin, and it will release the drug to be absorbed through the skin and can reach even to the systemic circulation and produce systemic effect. We can design dosage forms that will not release the drug immediately, but can release the drug at a sustained rate or delayed rate. So it will have a long duration of effect. So we can use excipients to control drug release rate. For example, controlled release tablets, transdermal patches, or some injections also, especially those oily injections that are injected, especially intramuscular in the muscles. They release the drugs at slow rates, so they have longer duration of effect. So this will be more convenient for the patient. Instead of taking 3-4 injections per day, they can take only, or tablets, they can take only one injection or one tablet per day. Some long acting injections can be given even every month. So instead of taking the patient every day, causing pain for the patient by daily injections, and just give one long acting injection that can be taken every month. So that is much more convenient for the patient. So in summary, for the previous, you want to make a drug product, then you have two main components: active pharmaceutical ingredient or drug substance. The dosage form or the drug product may contain one API or a combination of APIs. Sometimes you take flu tablets or capsules containing decongestant, antihistamine, and probably antipyretic and painkiller, analgesic, right? And it will contain also excipient or excipients, maybe more than one excipient, and this is most of the case. We use more than one excipient in the dosage form. And excipient, as you mentioned, is a substance that doesn't have pharmacological effect. They are other than the non-medicinal substance. It is excipients are substances other than the active pharmaceutical ingredient have been appropriately evaluated for safety and intentionally included in a drug delivery system. Okay? So together they make the drug product. So active pharmaceutical ingredient, as you mentioned, is the pharmaceutical of the pharmaceutical product is responsible for its pharmacological activity. We call it the medicament, we call it drug substance, we call it active ingredient. They are all synonymous. Okay? But when it comes to excipients, as you mentioned here, they should be evaluated for safety. So if somebody invented a new excipient for certain functionality, then Some of the requirements of the approval of this excipient to be used is to evaluate its safety. Once proven to be safe, then they can be approved and can be used. Second part of the statement is they are intentionally included in a drug delivery system. Intentionally means we add them, select them, add them by certain ratio or amount, mix them in a certain way to produce or to do or perform certain function in the formulation. Function, we will mention them in the coming slides, different functions produced by different excipients, and we will give examples. Once we prepare the drug product, then we have the pharmaceutical drug product. This is the finished dosage form. Capsule, for example, solutions, tablets, suppositories, solutions for injections, powder for injections, and so on. They creams, ointments, etc. They contain, it is a finished drug product, is a finished dosage form that contains active drug ingredient in association with excipients that make up the vehicle or formulation matrix. Okay. So formulation matrix is altogether drug and the excipients. Okay. So regarding excipients, any regulations? Of course, there are. We mentioned in the previous slide about the safety. So there are regulatory guidelines, and these guidelines always changing, improving. Depends on the growth of the body of knowledge about excipients and their effects and their degradation products and so on. So regulations of excipients showed steady growth in USA starting from 1906. The thing or the incident that provoked the growth of these formulations is a disaster that happened with sulfanilamide elixir. Used diethylene glycol as a solvent, and this one is a toxic solvent. It caused death of about 107 people as a result of the use of this toxic inactive ingredient or toxic solvent, diethylene glycol. So this incident provoked the regulatory bodies and dramatized the need to establish drug safety before marketing. So they put a requirement that the safety of drug products should be proven before marketing the drugs. And this provided the impetus to pass the pending. There was a pending law at this time, not passed yet, so provided the impetus to pass the pending Federal Food, Drug and Cosmetic Act 1938. So there are requirements or conditions for the use of excipients in the dosage formulation and stipulated by the regulatory guidelines from different countries, whether from USA, Europe, or Japan, or other countries. They are all common between these regulatory guidelines. So these requirements is number one, excipient will not have any harmful or toxicological effect. No harmful or toxicological effect. Should has a good stability with no drug excipient incompatibility. Doesn't cause physical or chemical incompatibility by any impurities in the excipient. So the excipient should be stable, doesn't degrade, doesn't produce byproducts, we call it degradation byproducts that will interact with the active pharmaceutical ingredient. So when we say incompatible means it will interact. When we say compatible means it doesn't interact, doesn't affect the API. Physical and chemical compatibilities. In general, most of them they are actions, the physical, but you can see there will be a change in color, there will be precipitation. So we call it physical. In this case, the change can be seen due to interaction between the ingredient, the API, and the excipient. Then we call it physical incompatibility. It is just a chemical reaction that you cannot see, but you can only know by analysis of your drug product. Then we call it chemical incompatibility. What is the problem of incompatibilities? It may affect the efficacy of drug products. Okay. Number three, there should be no interference in quality validation and analytical tests. For example, one of the quality control tests you perform on your drug product is to know the drug content, how much drug in this drug product, and how much is it compared to the labeled content. So, for example, the labeled content is that there should be 200 milligrams of drug in each 5 ml of the drug product. So you take samples of 5 ml of the drug product and then you analyze them and see what's the drug content in them. Of the requirements that the method of analysis you perform, excipients should not interfere. Means the result you get should be accurate irrespective of the amount of excipients. So because if the excipients will interfere, they may give you some higher results. Okay? So your result may be that content is 100% of the API, contains exactly the 200 milligrams, but actually the results are wrong and it was partly due to the excipients, but your content, for example, was only 150. So this is not a suitable analysis method. So usually in the evaluation of analysis methods also, you should prove that excipients will not interfere with the results. There should be no instability or incompatibility with the primary packaging material. Primary packaging material is the packaging material that will come into contact with the drug product. So, for example, you may have a syrup bottle, this one inside a carton box. The drug product is a syrup. So the syrup is inside the bottle. Bottle is in contact with the syrup. So the bottle is called primary packaging. Material of the bottle, glass, for example. So glass is your primary packaging material. The carton box outside is a secondary packaging material. Okay? Be physiologically inert, so it doesn't produce any pharmacological effect, doesn't produce side effects. Should has no unacceptable microbiological burden. So one of the quality control tests produced carried out on excipients is microbiological tests, and they should conform the required specifications when it comes to the microbiological count, or sterility test if it is for production of a sterile product. It should be satisfaction of regulatory issues and guidelines in all countries where the product is to be marketed. So this excipient may have monograph in British Pharmacopoeia, US Pharmacopoeia, Japanese Pharmacopoeia, and so on. So it depends on where do you want to use the country, in which country you want to use the excipient. Then the specifications of that excipient must be following or must conform with the specifications of the compendia or pharmacopoeia of that. Okay, for example, here in Malaysia, we don't have Malaysian pharmacopoeia. So if you want to use an excipient in Malaysia, so which guideline you should follow, then you should see the drug regulatory body. They may tell you that, okay, it is okay. We usually follow BP, for example, British Pharmacopoeia. It's okay to just follow British Pharmacopoeia or USP. Then in this case, you can just do the test and ensure that quality control tests and ensure that excipient is conforming to the guideline approved, the pharmacopoeia approved here in Malaysia. Okay, should have satisfaction for environmental issues, means it doesn't cause pollution to the environment. There should be ease of accessibility, distribution, and economical cost. Means I want to use materials that are not having shortage in the market, but available all the time. I can buy the amount I need when I want, so easily accessible. I have a local distributor here in Malaysia, so I directly can provide for me. It will take short time to get the amount I need than to contact an overseas supplier, and then shipment overseas will take more time. It will take time for approval to pass here to Malaysia and so on. And also should have the ease of economical cost, means it's not very costly. So at the end, I can produce a drug product with reasonable cost. So these excipients usually carry different names. Several names may exist for the same excipient. So how I'm going to use the name of the excipient? Each country has its own regulations. When it comes to US, the regulatory body in US, which is FDA, is requiring excipient nomenclature to follow certain um, precedence or order of precedence. And Malaysia also have similar regulations. So for labeling of pharmaceutical products, you write on the brochure or the package of the pharmaceutical product, the contents, write the active ingredient usually, and many companies, they also write the inactive ingredients. They write the excipients. So which name you are going to use for the excipient when it have different names? First, you should look for the name used in the pharmacopoeia, USP or national formula. If there is a name for the excipient there, you should use this USP name. You cannot find it monograph for it in the pharmacopoeia, then you can go for US adopted names and USP dictionary of drug names. This one, this issue. If no name, no monograph for this excipient, you can use International Osmotic Ingredient Dictionary and Handbook. Name is in this CTFA. If no name for it there, if it's not listed there, then we can try to use Food Chemicals Codex, FCC. Okay. Usually, many excipients are commonly used in food, also not only used in medicine. For example, you have starch. Starch is used to cook food, to mix many types of food. At the same time, used in tablet making, for example. Okay, so you may find it also in the Food Chemicals Codex. So now, if I want to find information about excipients, where can I find such information? And there are two different types of, or two main categories of information I may be searching for. I want to know about the excipient itself. What is the name? What is the molecular formula? What is the chemical structure? What are the different grades available in the market? What are the physical properties? What are the uses? In which ratios can I use them? And just want to know information about it, more information. Then your preferred reference should be the Handbook of Pharmaceutical Excipients. Okay, so here I'm not talking about quality control, not talking about only the regulatory requirements, but at the beginning, as an R&D or drug development specialist, when I want to know more information about excipient I'm going to use in my formulation, then I should use the Handbook of Pharmaceutical Excipients. So it presents monographs on more than 300 excipients used in dosage form preparation. And it's not just 300 excipients, because one excipient may be available in different grades. So different viscosity grades, for example, different particle sizes, whatever, they will all be listed under one excipient. It is same structure, same molecular formula. Okay? This difference in physical characteristics may cause difference also in their performance, difference in their use. We will give examples later on. So this book, the Handbook of Pharmaceutical Excipients, so as I told you, mainly you will use it as a drug development specialist or R&D pharmacist to find more information about excipients. Okay, it has monographs for more than 300 excipients. With each issue, you can find more excipients listed in that book. We mainly have information about non-proprietary chemical and commercial names, empirical and chemical formulas, molecular weight, pharmaceutical specifications. For example, should not include heavy metals more than this amount, okay, and so on. We will show an example. Chemical and physical properties, incompatibilities, and interactions with other excipients and drug substances. So you can collect this information first and review before you decide whether it is suitable to include it in the dosage form you want to develop or no. It also have information about regulatory status, safety, stability, handling information. Regulatory status means For example, it's approved by USP, it's approved by British Pharmacopoeia or no. What are the characteristics required by this pharmacopoeia? And then it will have information also about applications in pharmaceutical formulation or pharmaceutical technology. What's the use? Okay, what is the use? So this is an example. For example, you want to search for an excipient like hypromellose. Okay? So I search for hypromellose in this book, and then I find that hypromellose monograph is in page, for example, 326. I go to page 326. This is the kind of information I can find about this excipient hypromellose. Number one, what are the non-proprietary names? So you can find here in British Pharmacopoeia (BB). This is the pharmacopoeia issued by UK. Name is hypromellose. In Japanese Pharmacopoeia (JP), same name. In European Pharmacopoeia, same name. In USP, same name. As we say, usually it may have several names, many of excipients. So it will also give you the synonyms. Okay. So you have here the other names: penicillin, HPC, 464, hydroxypropyl methylcellulose, HPMC, hypromellosum, ethocell, methylcellulose, propyl ethocell or methylcellulose propyl glycol ether, and so on. So you have a lot of synonyms that are used for the same material. It gives also the chemical name, CAS registry number, cellulose hydroxypropyl methyl ether, and this is the code, the CAS number. If you want to find supplier, we usually search by this number. It gives you the empirical formula and molecular weight. And this is the structural formula. Functional category. It gives you the information about the functional category. It is bioadhesive material used as coating agent, controlled release agent, dispersing agent, solution enhancer, emulsifying agent, emulsion stabilizer, extended release agent, film forming agent, okay? And foaming agent, granulation aid, modified release agent, and so on. A lot of functional categories. But actually, it is available in several grades, different from each other in viscosity, and the use depends on the different grades of viscosity have different uses. Okay. Applications in pharmaceutical formulation or technology, as you can see, it is widely used in oral, ophthalmic, nasal, and topical pharmaceutical formulations. Oral products, it is primarily used as a tablet binder. Film coating is a component of the coat of the tablet. Okay, as a matrix, if we are producing coated tablet, then the film or the layer that form the coat around the tablet will contain this hypromellose as a main ingredient. It's used as a matrix for use in extended release tablet formulation. Concentrations between 2% and 5% may be used as a binder in either wet or dry granulation processes. High viscosity grades may be used to retard the release of drugs. A matrix at a level of 10 to 80% with weight in tablets and capsules, and so on. So it gives you idea about different uses and even the proportion to the amount that should be used for each application. Okay? Gives you a description. How is the appearance of this material? The specifications, pharmacopoeia specifications. You can see the tests here. Differences between different pharmacopoeias in Japanese and Pharmacopoeia Europaea and the USP. They may be similar in some tests, and there may be some differences in some tests. Okay. So here, pharmacopoeia tests identification, then appearance of solution with pH, the pH of the solution, and apparent viscosity, loss on drying, residual on ignition, and so on. Heavy metals, methoxy content, hydroxypropoxy content. Okay? So these are the pharmacopoeian requirements. Chemical properties after this. Acidity or alkalinity, what is the pH usually of? Then it gives you the pH for 2% weight per weight aqueous solution. Ash, once you burn, how much ash will remain? Auto ignition temperature, density, melting point. These are physical characteristics, and then specific gravity, solubility, and so on, viscosity. Okay? Then gives you also information about stability and the suitable storage conditions. Gives you the different grades available in the market. You have, so we call it methocell, as we mentioned, one of the names, other than hypromellose, is methocell. So there are different grades of methocell. They have different viscosity values. Okay. So what is the name of these grades in the pharmacopoeia? And then what is the viscosity in millipascals for each grade? Usually for 2% weight per volume aqueous solution of this material. It gives information about incompatibilities. So if your drug, once you review this information, you can exclude active ingredients of excipients, other excipients that may have incompatibility with this one. So if it is suitable to use with the other with your active ingredient, you can use. If it's not suitable, then you try to find alternative to use in the formulation. It gives you information about method of manufacture, safety, handling precautions, regulatory status. So here it's mentioned that GRAS listed, accepted for use as a food additive in Europe, included in the FDA and active ingredient database. So it's approved in Europe and FDA. What are the related substances? So either degradation product or byproducts of the reaction of synthesis of this material, and some comments, and then the reference is the source of this information. Okay, so last year I gave a very simple question. For example, list the information that you can find in the monograph, excipient monograph, in the handbook of pharmaceutical excipients. Is it a difficult question? No, many of the previous batch did not answer the question properly. Just enough to mention the title of each section. That's all. Okay. And if you know some examples or you are asked to give examples, it's easy. If you understand what is the meaning of this title of the section, then you can give examples. Okay. From regulatory point of view, if you are, for example, a quality control or quality assurance staff, so you want to, for example, write the document about the SOPs for the quality control tests that you should perform on the excipients once you receive them from the supplier, and to ensure that they qualify so that you can use them in the production. Then you can, your reference is USP monograph. Okay, why it should be USP monograph, not this handbook of pharmaceutical excipients? In a handbook of pharmaceutical excipients, still the tests are given, right? The required specifications are given, but the procedure is not given. But the pharmacopoeia will give you the required tests and the specifications, the test results that you should get. Also, it will give you the procedure for testing. Give you the procedure for testing. So in the USP, you will have also a national formulary and other pharmacopoeias as well. USP for the United States. You can find in the European pharmacopoeia and British pharmacopoeia, Japanese pharmacopoeia. Also, you can find monographs for excipients. So the monograph will have title, which is the excipient name. This is mainly the title of the excipient. Okay. So it will contain then a list of tests that you should perform on the excipients. Some of them are general tests, some of them are specific tests that should be applied or performed or conducted to characterize the excipient, to test the excipient. So some of these tests, the specific tests, will be a suitable stability indicating assay method or an accompanying procedure to identify impurities that can demonstrate stability. So stability indicating assay methods usually mainly to identify or to test the content, you can say the purity or the strength of the excipient. Then also it may contain a procedure to quantify or to detect the limit of degradation product of that or impurities of that excipient. Okay. It will have a name. It will describe this monograph. The next section will be description, physical properties, for example, white powder, off-white powder, for example. Okay, having certain smell, whatever, to describe it physically. And they will give the identification tests. How can you identify this material? So qualitative tests to identify it. Then quantitative tests for assay and assay of impurities. And it will give information about packaging and storage conditions suitable for this. And any specific tests needed to better describe and control an excipient. For example, microbial limit test, pH, and so on. Let's see an example, sample monograph of the pharmacopoeia. So we go to the same material. You can see it now. You can see this PDF document, hypromellose. Can you see it? Okay, so if we have a look on hypromellose, you can find, previously we showed the monograph of hypromellose, the handbook of pharmaceutical excipients. Now we show it in the pharmacopoeia. Pharmacopoeia is not concerned with giving you too much information about this excipient. They only want, or they are concerned with giving you information about the requirements for this excipient and how to perform the quality control tests. So here it gives you information about definition, what is it, and then how to identify it, identification tests, and then assay tests. And then as you can see, test for impurities, okay, residue on ignition, for example, heavy metals, and then some specific tests. You should test the pH, viscosity, using the capillary method, rotational viscometer, okay. Some additional requirements, packaging and storage and labeling. So pharmacopoeia will all mainly concerned with providing information about regulatory requirements. This should be the specification of this drug. Physical properties 1, 2, 3 must be like this. You should identify, do some tests to identify your material that it is really hypromellose. You should do some assay test. You should test some pH, solubility, whatever. Okay. Then only if your material conform to all these tests, then you can use it in your production. If it doesn't conform, then you cannot use it. Understand? So this is the difference between the Handbook of Pharmaceutical Excipients and the Pharmacopoeia. So the Handbook of Pharmaceutical Excipient will be required when you are in a planning stage, you are developing a dosage form, you want to collect more information about your excipient as much as you can. So it is, you can say it is a tertiary information reference that contain most important information about your material. Then if you want to write the SOPs for the tests you'll do in the quality control department, verify that the supplier, the supplied material or supplied excipient conform to the specifications, and you can use it in your production. Then you refer to the pharmacopoeia. You can find required tests, the results, accepted results, and the procedure. It is written there. Okay. So then we can have a 5 minutes break before we can proceed to the second part of the lecture. Okay. So let's resume our lecture with the excipients functional categories. We will give examples for different functions the excipients can perform, and we give examples for such categories. What are the excipients that are classified under this category? When we talk about excipients functional category, we mean a qualitative classification or term that describes the purpose or role of an excipient in a drug product. What is the function? What is the role of the excipient in the drug product? So you have different functions excipients can perform. Some excipients can be used, for example, like acidifying agents to keep the pH at acidic range at a value that is suitable for the stability of the drug product. Some drugs may be more stable in acidic medium, so we use acidifying agent. Several acids can be used like citric acid, acetic acid, fumaric acid, HCl, nitric acid, and so on. We may have an alkalinizing agent. Some drugs have another requirement. They may be more stable in alkaline medium. So we then keep the pH of the medium alkaline. So we can use some alkaline substances like ammonia solution, ammonium carbonate, diethanolamine, monoethanolamine, borate, carbonate, trolamine, etc. Okay. Some materials can be added for the formulation to act as an adsorbent. They are agents capable of holding other materials onto the surface by physical or chemical means, like powdered cellulose, like activated charcoal. Some are used for air displacement. If the drug is easily oxidated, so then it will be packed in a hermetically sealed container, means doesn't allow the passage of air. And it is not 100% full. There is a space inside the container. We replace the air in this space with a gas that doesn't react the drug, like nitrogen or carbon dioxide. Okay, so we use it for air displacement. Some excipients function is to be an aerosol propellant. This is an agent responsible for developing the pressure within an aerosol container and expelling the product when the valve is opened. In aerosols, there is a compressed gas usually, and this compressed gas is liquefied by compression. So inside the container, most of it may be in a liquid form, and there is a gas also in the space above the liquid, and this causes certain pressure inside the aerosol container. Once you just press the valve, it will release the gas
outside with this gas will cause pressure that will release the liquid outside, and this liquid contains the drug in its matrix. Okay, so several gases can be used like carbon dioxide and fluoromethanes or fluoroethane. Okay, difluoromethane, dichlorotetrafluoroethane, trichloromonofluoromethane. They are used in this way. Some are used as preservatives, like parabens, for example, methyl paraben, ethyl paraben, butyl paraben, and so on, and benzoic acid and sodium benzoate. They have antimicrobial effect. They have effect that doesn't allow the growth of fungi and bacteria as well, but their effect on fungi is stronger. That's why we classify here under antifungal preservatives. So they prevent the microbial growth in your formulation, so it will be stable and will be effective and will not harm the user during the shelf life. Okay. Some preservatives mainly have antimicrobial effect, whether antibacterial or antifungal, like benzalkonium chloride. So they are used in liquid and semi-solid preparations to prevent the growth of microorganisms. Okay, so again, parabens, which are derivatives of, they are esters of para-hydroxybenzoic acid, and the benzoate. Usually, they have a stronger antifungal effect compared to the antibacterial effect, but still they have antibacterial effect as well. Okay. Some materials may be used as antioxidants. Your drug product may be prone to oxidation, so to prevent the destruction of your drug during the shelf life by oxidation process, you can add some antioxidants in the formulation so they will stabilize the drug. They will prevent the deterioration of preparations by oxidation. And they have reducing ability usually, so there are a lot of materials can be used. Some of them are water soluble, some of them are oil soluble. So it depends on your formulation. What is the medium there? It is aqueous medium or oil medium. You can use the appropriate agent, whether water or oil soluble. Ascorbic acid is vitamin C is one of the examples. Hydroxyanisole, hydroxytoluene derivatives, bisulfites and metabisulfite also, they are commonly used as antioxidants. We use some buffering agents, which is composed, as you know, from weak acid and conjugate salt. This in order to fix the pH of the formulation and to resist the change in the pH upon addition of acid or alkali. And this pH will be adjusted to be suitable for the stability of the drug product and for the good performance of the formulation. So many salts can be used together with the weak acid. Chelating agents may be used in some formulations, and their function is mainly to bind metals and to form soluble complexes, so they will not precipitate upon binding this metal, so they will not cause any physical changes in the formulation. Okay. And why do we use them? We use them in case where the heavy metals will cause instability of the drug product. Okay. For some drug products, metals may promote instability. So to keep the drug product stable and effective during the shelf life, the formulation must contain chelating agent, and examples are edetic acid, or that we can call it ethylene diamine tetraacetic acid, or shortly we can call it EDTA, ethylene diamine tetraacetic acid, or edetic acid, or its salt, disodium edetate. We can use some coloring agents or colorants that to impart a color whether to the liquid or the solid. Okay, so they can be added to syrups, to solutions, to give them a liquid, to improve the appearance. They can be also included in the coat of tablets. They can be in the shell material of capsules to give them the required color. And there are a lot of coloring materials that have different colors. Sometimes can be used alone or can be mixed together to give a certain color grade. There are clarifying agents used as filtering aids for its adsorbent qualities like bentonite. Emulsifying agents, they are, when we want to make emulsion, then we can use emulsifying agent in the formulation. So they promote and maintain the dispersion of finely subdivided particles of liquid in a vehicle in which it is immiscible. Emulsion is usually when you want to mix oil with water. If you put oil and water together, you shake, whatever, for how long you shake, then still they will separate into two different layers. One layer is oil, one layer is water. But if you add emulsifying agent, whether natural or these substances or synthetic like polyethylene 50 stearates or butyl monooleate, some of them are natural like acacia and some are synthetic. You add emulsifying agent, then you will get one of them will be dispersed in the other as a fine droplets that you cannot see with the naked eyes, same like the milk. Okay, so emulsifying agents are used to make emulsions and used also to make a cream. A cream is also an emulsion, but we add a thickening material that will make it thick, not liquid like the milk. Okay. There may have encapsulating agent. This is a material used to make the capsule shell, like gelatin. Most of the capsules in the market made from gelatin. Some of them are made from hypromellose, just small portion. So they form the thin shell to enclose the drug for ease of administration. So we make the capsule from gelatin. Inside the capsule, we put the formulation that include the API. Some materials are used as flavorant just to give certain flavor, smell, and taste, like anise oil, cinnamon oil, cocoa, menthol, orange oil, peppermint oil, vanillin, and so on. Depends on the flavor you want to give. Sometimes in the formulation we use humectant. Humectant is a substance that will absorb water and prevent drying of the preparation. So we use it mainly in ointments and creams. So during the shelf life, anytime you open the tube, you will not find that your cream or ointment become hard, but still have the same consistency. Like glycerin, propylene glycol, and sorbitol, they are used as humectants, especially in ointments and creams. Levigating agent, this used an intervening agent to reduce the particle size of a powder by grinding, usually in a mortar. So you have a powder you want to grind in a mortar. If you grind the powder as it is, it will be difficult to grind it all because some will be by the effect of the pestle of the mortar. They may form clusters, they may be compact, but you can use an agent that intervene between the mortar and pestle and the powder. You can use some oily material or thick material like mineral oil, like glycerin, propylene glycol. You add to the powder and then you keep grinding. In this case, you will not get any crusts or you will not get any compacted powder, but they will be distributed in the liquid and will be ground by or will be grinded by the effect of the friction between the mortar and the pestle. If you want to make ointment, then we should include the API in an ointment base, and this may be oily and may be non-oily materials. There are a lot of materials like lanolin, naturally taken from the sheep, hydrophilic ointment, like polyethylene glycol. They may use some materials derived, oily materials derived from oil or petroleum, like petrolatum and hydrophilic petrolatum. White ointment, yellow ointment, rosewater ointment, they are all different ointment bases. You will discuss in details in the lecture about ointments and creams. Some materials are used as plasticizer, and from the name plasticizer, like plastic. What is the property of plastic? You can twist the plastic, it will not break, right? So you make it more elastic. So if we put plasticizer in your matrix, it will make it not easily breakable. Okay. So it is usually we add the plasticizer to the film coating materials. You want to coat tablets with a thin film of certain material. We add plasticizer to it like diethyl phthalate or glycerine. This material usually will prevent cracking of the coat upon storage and upon shipping the tablet. Okay, so when you buy a film coated tablet, especially colored tablets, this is definitely have a coat that give the color to the tablet. So you find always that it is not, there is no crack or break on the surface. Why? Because the material that form the coating containing a plasticizer. So make it more pliable, enhance the spread of coat over tablets also, or beads and granules. Whether you coat tablets, you coat beads, you coat granules, spread of the coating material when you spray it will be easy and then it will not break with storage. Solvent, a lot of solvents can be used to make the liquid formulations specifically. The solvents, there may be solvents, there may be co-solvents. Solvents may be aqueous matter like water, purified water, sterile water for injection if you want to make injections, sterile water for irrigation, and so on. And usually we use it to dissolve the water soluble materials or drugs. And some preparations may be containing oil soluble drugs, so they may be dissolved in oils, mineral oil, oleic acid, peanut oil, cottonseed oil, or corn oil. Like some injections. Some injections are oily material and mostly they are for sustained release. Okay, so we just dissolve the oil soluble drug in oil, we give it as injection. Sometimes you have oil soluble drug, but you want to make aqueous formulation. So how to dissolve it? You can use co-solvent like alcohol. And sometimes the solvent is mainly alcohol, but sometimes you use the alcohol as co-solvent. So you mix alcohol with water, so they will be able to dissolve both water soluble and water and oil soluble drugs. Some materials are used as stiffening agents. As we mentioned in the previous slide about emulsion, emulsion is, for example, oil and water emulsion. You disperse oil in water as a fine droplets that you cannot see by naked eyes, but you can see under microscope, for example. It will have a white appearance like the milk. Okay, so this is just an emulsion. But if you want to make it as a cream, the cream is thick, the consistency is thicker. So how to make it thick, how to convert it from emulsion to cream, you have to add a stiffening or thickening agent like cetyl alcohol, stearyl alcohol, microcrystalline wax, white wax, yellow wax, and so on. So it can increase the thickening or hardness of a preparation, usually used for ointments or creams. So it can be used for creams as I explained. For ointments also, you can use different proportions of different types of bases. Some of these bases may be thin or light, some may be thick. You mix different proportions to get the consistency you want. Okay. Some materials are used to make suppository bases, so they are vehicles for suppositories. We can mix the API that is intended to be delivered as a suppository with these materials. We call them suppository base, like cocoa butter, very commonly used, or polyethylene glycols, different grades. Surfactants, they are used to make surfactants. Actually, they have different functions. They can be used as emulsifying agents. Depends on something we call it HLB, hydrophilic, lipophilic balance. So it depends on the value. There may be used as emulsifying agents or wetting agents or detergents. Okay, and some of them are synthetic, some of them are natural. Here, all listed here are synthetic. Suspending agents, they are usually thick materials. If you make a suspension, usually you shake the suspension and then all the suspended material will be suspended in the vehicle, whether aqueous or non-aqueous vehicle. Okay, but then if you leave it for a time, what will happen? It will precipitate, and this is not suitable. If you want to take an accurate dose, you shake. You want it to be suspended for a long time, enough to take the dose. Okay, So you have to add in the formulation a suspending agent. What is the suspending agent? It is a thickening agent. It is a viscosity increasing agent to make the vehicle thick enough so that it will not allow rapid precipitation of the suspended particles. Okay, so it will be used to reduce the sedimentation rate of particles in a vehicle in which they are not soluble. Suspension may be formulated for oral, parenteral, ophthalmic, topical, or other routes. You can find a lot of types of drug products in the form of suspension. Some are injections, some are eye drops, some are for oral administration, okay, and some for topical administration, like calamine lotion. It is for application onto the skin. So we have a lot of materials. The common between them, most of them are polymers, except kaolin here is not polymer. Yeah, okay, kaolin is something like, kaolin, it's like mud. But the rest are polymers, so they have high viscosity. They are thickening material once you add them to, especially they are added to aqueous. Bentonite also is same like kaolin, it's a mineral origin. The rest are like agar, carbomar, cellulose, and so on. They are polymers and they are thick. Once you add them to aqueous vehicle, they will increase its viscosity and make it thick. In some formulations, especially for oral, mainly, of course, for oral administration, you may need to add sweetening agent to improve the taste, to mask the unpleasant taste of the drug. So they are used to impart sweetness to a preparation, like some synthetic materials like aspartame or saccharin, and some are natural like glycerin, dextrose, mannitol, sorbitol, and sucrose. Okay. In tablet production, we use adherents, binders, fillers, or diluents, and disintegrants. These are the main components of tablets. So what is the function? Number one, the diluent. This will make most of the amount, just this to increase the weight of the tablet to the required weight. It's just an inert filler to create the desired bulk. It should also cause the flow properties and compression characteristics of tablets and capsule. It largely affect the flow properties, compression characteristics of tablets and capsule because it is the major component. Okay, so actually mainly I make a tablet. The main ingredients are two ingredients: active ingredient and diluent. So I'm making 5 mg tablets, and I want the tablet weight to be, for example, 200 milligrams. So most of the remaining of the 200 milligrams after the 5 mg will be the diluent, or we call it filler. So we call it either diluent or we call it filler. Okay, so they are inert material. They don't have any pharmacological effect. You can use some natural materials like dibasic calcium phosphate. This is mineral origin. Organic material like lactose, mannitol. Synthetic material like microcrystalline cellulose from cellulose. Natural material like microcrystalline cellulose. Powdered cellulose, precipitated calcium, mineral origin, calcium carbonate, sorbitol, and starch. They are all can be used as fillers. But then I have to add some important ingredients for the tablet making, to give it some characteristics. Number one, I want to add binders. Tablets are made by compression. I want to be sure that after compression they will not break again. So there must be a material that it acts like a glue, and they keep the tablet components bound to each other. Okay, the particles bound to each other. So they cause adhesion of powder particles in tablet granulation. And these substances, as I told you, they act like a glue. Most of them, if you mix with water, they increase the viscosity and they also may be sticky. Okay, they have some stickiness. And then I want to be sure that once this one is swallowed and come into contact with gastric fluids, it can break easily. So I have to add another material that will cause tablet disintegration. Disintegration means breaking up. Okay, So they used in the solid forms to promote disruption of the mass into smaller particles, more readily dispersed or dissolved. So I use them in the solid dosage forms, tablets, to promote disruption of the mass into smaller particles, more readily dispersed or dissolved. So it will cause rapid dissolution of the drug once comes into the GIT. So a common sense, tablet disintegrant will be used in immediate release tablet, but they will not be used in sustained release tablets. Because for sustained release tablets, I want the tablet to dissolve slowly, so I will not add any disintegrant. But if I want to make immediate release tablet, then I have to add disintegrant so that once coming into the stomach, it will break up quickly. Or if it is enteric coated, once the coat dissolves in the intestine, it will break up quickly and it will release all the particles, small particles that will dissolve quickly. The drug will dissolve quickly and then will be absorbed. The examples are alginic acid, polacrylene sodium, sodium alginate, sodium starch glycolate, and starch. How they are going to produce this function, the property here that they swell. Once they absorb water, they swell. So once they swell, they make a force that will bring particles away from each other. Okay? So binders act like glue, but disintegrants usually they're swelling agents. They will swell and then they will cause or they push the particles away from each other and cause disintegration of the tablet. Okay? Additional ingredient that are used in tablets is the anti-adherents. Tablet making is by a machine that contains a punch and die. The punch will exert a pressure on the tablet powder, on the powder. Once compression applied, it will be compressed into a tablet. This part is moving up and down, making pressure going up again. And then another powder coming under it, it will press it and so on. So there is a cycle, you have a circle, and then this circle in the machine rotating continuously, and this punch keeping going up and down, up and down. So I want to add a substance, we call it anti-adherent, that will prevent the tablet ingredients from sticking to the punch and die during production. I don't want the punch once going down and pressing the powder that the compressed tablet will stick to the punch and goes up with it. Okay, then it will interrupt my production process. I want the punch to press into a tablet and then the punch will go up alone and the tablet will be expelled from the machine. Okay? So I have to add some greasy material usually. Okay, so this material is usually greasy material, something like have some lubricating properties like magnesium stearate. Stearate is an oily material, so the salt magnesium stearate, stearic acid can be used. Okay, talc can be used, which also have some greasiness, and they are working as tablet anti-adherent. Some of the materials used are direct compression grade. So from these fillers themselves, like dibasic calcium phosphate or like lactose, it comes in different grades. It may come as fine powder or may come as coarse particles. If it is coarse particles, then it is direct compression grade, like dibasic calcium phosphate ditab, like dicompression grade of lactose. The difference between direct compression grade and normal grade is the particle size. They are usually coerced, so they have a good flow. If we want to make coated tablets, then many materials can be used in making the coat. Depends, there are different types of coats. Some coats are to protect from humidity and atmospheric oxygen. Some coats are to give a sweet taste, like sugar coating. And some coats to protect from the humidity and oxygen, we call it film coating. If it is to protect from dissolution into the stomach, then we call it enteric coating. So different types of coating We use for different types, we use different materials. Um, so we may use sugar coating just to mask the taste or odor. So in this case, we just sugar, we make a coat on the tablet made from glucose or sucrose. And we want to give for aesthetic purposes, we want to give some nice color or whatever, we can use film coating. So in this case, it will, and also they protect from humidity and atmospheric oxygen. So usually polymers are used together with plasticizer. And then we have another material also like polyethylene glycol to cause rapid dissolution when it comes into contact with water. If I wanted to keep intact in the stomach, not to dissolve in acidic pH of the stomach and just dissolve once reached to the intestine, then I make enteric coating. I use substances like phthalates usually, or shellac, cellulose acetate phthalate. There are different phthalates that can be used for enteric coating, and shellac, this is a natural material from bugs, dissolved in alcohol in a concentration of 35%, then I spray it in the machine, in the coating machine, and the solvent will evaporate, and then the shellac will form a layer around the tablet. This is, we call it pharmaceutical glaze. Pharmaceutical glaze is the solution to the coating solution. Okay. In tablet making also I use something called tablet glidant, and the glidant will allow, it's lubricating also, but not to lubricate between the tablet and the punch, but to lubricate between the particles of the tablets themselves. So used in tablet and capsule formulations, it will improve the flow properties of powder mixture. Okay, the powder that you mix together and you want to compress to make tablet, or you want to fill into capsule shell. You can add a glidant that will make like lubricant between them, prevent their stickiness together, and they can flow better, like colloidal silica or cornstarch or talc also can be used as glidant. We mentioned about lubricant. These are the examples other than stearate. We can use tablet or capsule opacant to make it more opaque. So like titanium dioxide, usually it is added in the coating material. We can use tablet polishing agent to polish the tablets after production to give them a shiny appearance. Carnauba wax or white wax can be used for polishing. Tonicity agent can be used in liquid formulations to render the solution similar in osmotic characteristic to physiological fluid. Especially we use it for parenteral and ophthalmic solutions and in irrigation fluids that we use it to clean the wounds. Irrigation fluids are used to clean the wounds. So we adjust the tonicity to make the osmotic pressure similar to the blood. Okay. In this case, we call it isotonic, to make isotonic solutions. So sodium chloride is used to adjust the osmotic pressure and to make isotonic solutions. We can use some viscosity increasing agents in many formulations. Used to render preparations more resistant to flow. When we talk about liquid preparations, for example, used in suspensions to decrease the sedimentation rate, used in ophthalmic solutions to enhance contact time. Once you instill a drop of eye drop, it will remain in the eye, it will not be flow out of the eye quickly. And we use it to thicken topical creams. We use it for sustained release tablets, many uses. And most of them are thick materials and polymers. Okay. Last thing is the vehicle. This is a carrying agent used in formulating a variety of liquids for oral and parenteral administration. So in tablets or capsules, we call it diluent or filler. But when we talk about liquid preparation, then we call it, if we talk about semi-solid, we call it base. If we talk about liquid, we call it vehicle. Okay? Like, for example, oral liquids usually are aqueous, so the vehicle is water, or a solution of sugar. Can be hydroalcoholic, means mixture of water and alcohol, as I mentioned, to increase the solubility of lipid-soluble drugs. So we call it elixirs. Solutions for intravenous use are usually aqueous. We cannot give by IV an oily solution, but intramuscular injections can be aqueous or can be oleaginous. Okay, so again, three different names. When we talk about semi-solid preparations, like ointments, creams, suppositories, we call it base. Okay, the diluent or whatever, we call it base. When we talk about solid dosage forms like tablets and capsules, we call it diluent or filler. But when we talk about liquid dosage forms, we call it vehicle. Okay, so can be flavored vehicle or sweetened, like acacia syrup. Always when we say syrup, means there is sugar. It is a sweetened. Okay, when you say elixir, then there is alcohol. When you say elixir, then there is alcohol. Okay, so it can be flavored. Acacia syrup contains acacia. This is a thickening agent, suspending agent. Aromatic syrup is having aromatic flavor. Aromatic elixir means elixir that have aromatic flavor as well. Sherry syrup is syrup that flavored with sherry flavor. Cocoa syrup with cocoa flavor. Orange syrup with orange flavor. Just we say syrup, then it is a plain syrup. Sucrose in water. Sucrose in water. Vehicles may be oleaginous. We can use some oils, as we mentioned, in injections that intended for sustained release and given by intramuscular route. It may be sterile, like bacteriostatic sodium chloride injection. This is usually isotonic injection. Okay, then we come to the part of excipients characterization. What is the importance of excipient characterization? When we say characterization, what does it mean first? Characterization means I want to know the characteristics of the excipient. Okay, so what are the characteristics? The properties. What are the properties? Generally, we classify them into physical and chemical properties, right? Physical and chemical properties. So, when you talk about physical properties, for example, what is the color, what is the smell, what is the melting point, viscosity, okay, solubility, pH, and so on. Then you go to chemical properties. Then I want to know that, for example, the assay, degradation products, and so on. So what is the importance of excipients characterization? Why I have to know the characteristics or I make tests for the excipients? Because they will affect formulation stability and performance. Okay, they affect formulation stability and performance. I give you a simple example. We give examples of many thickening agents, and we said that thickening agents are also used as sustained release, used to make the sustained release tablets. So usually sustained release rate, the release rate of the drug will be dependent on the viscosity of the excipient, the viscosity of this polymer. Okay, this polymer will be available in different grades of viscosity. Different grades of viscosity, once made or put in the tablet in the same proportion, they will result in different release rates. So I have to determine, I study already the effect of viscosity of the polymer on the release rate, and I choose certain degree of viscosity. Then I have to know, before I use any batch, I have to determine the viscosity, so that I need to know if it will be okay, or I have to do some adjustment in order to achieve the release rate I want. This is the performance of my sustained release tablet. The main indicator for performance here is the drug release rate. Okay. Second thing, the drug substance can be susceptible to chemical and physical changes induced by the properties of the bulk excipient. We'll explain what are the properties of bulk excipient, like viscosity, for example, and so on. Implementation of quality by design product development strategies place an increased emphasis on detailed characterization of excipients to achieve robust formulation and process. So I gave the example. When I do formulation development, I have a target drug release rate. For example, I want 80% of the drug to be released after 12 hours from the tablet. Okay? So I try different grades of certain polymer, and then I study the relationship between the viscosity of the polymer and the release rate. This is what I call it quality by design. From the beginning, I do design. I do experimental design. I vary one parameter that may be very important and affecting the performance of my product. So here, the example I give, the parameter is viscosity of the sustained release of that drug retaining polymer that I use, and its effect on the performance criteria of the tablet, which is the drug release rate. So I have an experiment from the beginning. I have a range of viscosity I tested, and then I see within this range what is the effect on the drug release. Then I can predict the effect of variation of this quality of this parameter on the quality attribute of the product, which is drug release rate. Okay, so there is effect for this characteristic, which is viscosity of the polymer, drug retaining polymer, on my formulation performance. Okay, so then I have to test the viscosity before I can decide to use it or no. So I define a range of viscosity suitable to achieve the release rate I want. Once I receive this excipient or drug retaining polymer from the supplier, then I have to test the viscosity, and depending on that, I have to decide whether this is suitable to be used or cannot be used, okay, or need some adjustment. So in any formulation, as you see here, role and function of drug excipient to ensure quality, safety, and efficacy of the active ingredient. Okay, how to ensure, as we mentioned that in each dosage form we use excipient plus drug. They should be compatible with each other. They don't interact chemically or physically. So my product will be stable for the shelf life. Stable means will have required efficacy will not change, the safety will not change, and it has the required quality that will not change. These excipients, there are a lot of different types of excipients that can be used. As we mentioned in the dosage form, I may use some coloring agents, and the stability of the coloring agent affect the appearance and identity of the dosage form. So this dosage form is, from the beginning, the characteristic is, for example, it is pink. The appearance is pink, and I know it by its shape. There may be some print on it, and then the color. So if coloring agent is not stable during the period of shelf life, then it will affect the appearance and identification of the dosage form. Some are used for patient compliance, like flavoring and sweetening agents. So they must be stable. They must not interact with the active ingredients, so that they have the same characteristics during the shelf life of the product, and they will not affect patient compliance. Some are used for manufacturing, like, for example, carrier constituents, solvent, liquid vehicle, suppository base, diluent, propellant, etc. They are essential part of the dosage form. They must be there. For example, if your propellant escapes from the container of the aerosol, it will not act. Then you lost the efficacy, right? Suppository bases should not change, should not interact with the drug, so that the drug release from the suppository base will be the same. The performance of your product is the same. Your ointment, for example, base should not dry up during the manufacture, so that will not affect the performance of the product and also ease of application and so on. Some agents are added for processing aids like binders, glidants, enhancers. We mentioned about them already. They all should be stable, like emulsifiers and so on. How about if your emulsifier degraded? You will not have emulsion anymore, and your ingredients will separate into oil phase and water phase. Okay? In modified release dosage forms, we are using some materials, some polymers, coating materials for sustained release. They are important to ensure the performance of the product and to have the same quality. Disintegrants, wetting agents. Disintegrants will ease the disintegration of the tablet, so it will produce rapid effect. How about there is malfunction of disintegrants? Then the tablet will not disintegrate quickly, the drug will not dissolve quickly, and it will take longer time for the drug to be absorbed and to produce effect. And so on. We have materials to increase the stability. Chemical stabilizers like antioxidants or chelating agents. Physical stabilizers like viscosity enhancers or gelling agents. We have preservatives to prevent the microbial growth. We have buffers to modify the pH and to keep the drug stable. All these ingredients we should ensure that they are stable and produce the intended function during the storage period, and they should not interact with the drug so that they don't affect the drug efficacy. They don't cause the production of toxic degradation products, for example, or affect the performance of the product and cause rapid release that will cause toxicity, for example. Okay, and they will keep the quality of the product. Excipients are classified based on chemical structure into three main categories: organic, inorganic, and polymeric. They are, as we mentioned previously in the table, derived from natural sources or synthesized by chemically or by other means, by biotechnology. For example, if we talk what other means other than chemical means, I can tell you there can be a biotechnology product, simply. Okay. They range from simple, usually highly characterized organic or inorganic molecules to highly complex materials that are difficult to fully characterize, which are polymers. Usually polymers are highly complex materials and difficult to fully characterize. So inorganic, like some iron oxide as a pigment, calcium phosphate as a filler, inorganic. Organic, same like mannitol, sorbitol, sucrose, glycerine. Okay, we mentioned many of them in the previous tables. Polymeric, like mostly the thickening agent and drug retaining polymers like hypromellose, starch, okay, gelatin, and so on. They are polymers. Ketosane, they are polymeric materials. Okay. As we mentioned before, we give an example: compendial tests required for characterization of excipients. There is a monograph for each excipient, and there are tests that should be performed, but keep in mind there are a lot of variabilities among excipients. One excipient, as we mentioned previously when I discussed about the table of functional categories of excipients, I told you one excipient may be available in several grades, and I showed to you already in the hypromellose monograph in the handbook of pharmaceutical excipients. So one excipient may be available in several grades and supplied by different manufacturers. Different grades of the same excipient may vary in polymer molecular weight, degree of substitution, particle size distribution, and morphology. Different manufacturers often employ different synthetic schemes or isolation techniques, so we can have some slight differences in physical properties like melting temperature, crystallinity, loss on drying, particle size, and even in chemical profile. The trace impurities may be different from one manufacturer to another. Okay. So there are a lot of differences in manufacturing, as I told you. Excipient sources may be plant, animal, mineral, chemical synthesis, biotechnology sources. So they may vary from manufacturer to another. For example, magnesium stearate or stearic acid may be taken from plants, some of vegetable origin, and some suppliers will produce it from animals, including pork, okay, including porcine origin from the pigs. Okay, so materials, some materials also may be natural and some material may be synthetic, like alcohol. Alcohol may be produced by fermentation and may be produced synthetically. So you get it from one supplier, it may be from natural source, from fermentation, for example. It may be also from synthetic origin. Synthetic origin may have different byproducts compared to natural origin alcohol, isn't it? And so on. And then the excipient manufacturer, they have also difference in manufacturing process. Some of them are producing different batch by batch, and some may have continuous manufacturing. They may be different in the harvesting method, how to separate my material from the reaction medium, how to extract it. There may be some difference, little difference in chemistry. If I'm doing some derivatization for cellulose, for example, I'm doing HPMC, I'm making HPMC. So it is hydroxypropyl methyl cellulose. Okay, so I'm doing propylation and methylation for cellulose. So the degree of methylation and propylation may have some variation. There some be some agglomeration differences, and so it will affect the particle size. The size reduction also process after collection and harvesting and agglomeration then may be different from manufacturer to another, may result in different particle size. So these differences in processes and in source may result in differences in microscopic and macroscopic properties. They may result in differences in moisture content, crystallinity, dehydration, enthalpy of melting, glass transition temperature. They may result in difference in density, flowability, compressibility, viscosity, and surface area. And this variability can be source to source variability, means difference in these properties from manufacturer to another. When I say source to source variability, I mean different manufacturers. And there may be even lot to lot variability. Lot is batch. So actually I experienced it before. I'm using HPMC as the drug retaining polymer for sustained release tablet. When I bought my HPMC certain grade from the same source, the same manufacturer, they supplied it to me. The specification telling that the viscosity should be in the range of this value to this value. Okay, say for example 50 to 100 CPS. First batch they give to me is the viscosity, say 70. And then I develop my tablets. I determine the amount I should use to achieve certain drug release rate. Okay, then when I come to manufacturing, I ordered again from the same source, same company. Then when they supply it to me, say the viscosity, for example, is instead of 70 is 80 CPS. So does it affect the drug release rate? Yes. So it will change it. It will make it lower. So it will change the drug performance. So what to do? So that's why from the beginning you have to use quality by design. You use different grades of solubility, of viscosity, different manufacturers, and then you study the effect, and then you get your formula, how much you are going to use in the tablet. So you can adjust the content based on this formula. Okay? So we have lot to lot variability. So source to source means different manufacturers. Lot to lot variability means same manufacturer but different batches. Okay? So these variabilities in characteristics will affect the functionality of the excipient and will affect the performance of the excipient and the product as well. So it will affect the critical quality attribute of the final drug product. In this example I gave to you is the drug release rate. Okay? So critical quality attributes can be affected by excipient performance, by formulation variables, changing the amounts of ingredients in the formulation or changing the grades of the excipients in the formulation, by changing the API properties, physical properties even can affect. This time, if you use, for example, micronized versus non-micronized drug, for drugs that are not soluble in water, then they will affect the absorption. Micronized have better solubility and better dissolution properties and better and faster absorption, right? And process variable itself, process variables. Process variables, we are talking about the steps of processing. How long you are going to mix the powder together, how long you are going to um, what do you call it, going to do the granulation time for granulation of tablets, for example, emulsification, you want to produce emulsion, how long you are going to use the homogenizer to produce your emulsion, and so on. These are the process variables. Okay, so when you talk about process variables, means the processing steps, whether you will change in these steps or no. The time of the mixing, the time of homogenization, for example. You want to make syrup, the time of using the homogenizer to dissolve the sugar, for example, and then to dissolve the active ingredient and so on. These are all processing variables. Okay, so when it comes to the functionality and characterization, formulation scientists usually rely on numerous compendial excipient characterization, chemical and physical analysis, tests or testing methods, and develop novel methods to analyze key quality materials attributes of a formulation. So they may use compendial tests and may develop their own tests. There are general chapters in USP giving tales of testing methods that cover elements of the chemical and physical properties at various levels. Molecular level, for example, there are description of the use of NMR, INR, NIR, and UV spectrophotometry to identify the drug and to quantify it as well. Usually IR or NMR is used for identification. UV is mainly used for, can be used for identification, but mainly used for quantification. On particulates level, so we measure the particle size and the distribution of particle size. Use of optical microscopy for this issue also. And on the bulk level, bulk material, the measurement of viscosity, loss on drying, thermal analysis, and so on. Okay. These methods provide the core testing protocols for routine certification of materials for release specification. So here we talk about quality control. You are a quality control to release the drug or to allow to be used in the factory. You must do some quality control tests. And based on the results of the tests, if they fall within the specifications, they conform to specifications, you can release them. If not, then you reject them. So you perform the tests, you issue a certificate of analysis, and based on the results given in this certificate of analysis, it will show that whether this substance conform or doesn't conform, okay, to the tests. These tests are many, some are universal, means they are not specific to certain material. They can be applied to different materials, so they show generally how they are performed. For example, like if you want to make mass spectrophotometry, chromatography, thin layer chromatography, identification test as a general, how to determine the optical rotation and so on, and near infrared spectrophotometry, for example. So some are for description, some for identification, some are for assay. Okay. Chromatography is used for assay. If you go to this chapter, it will show you in general what is chromatography, how to do it generally, the process itself or the technique. But if you want to use chromatography to determine for assay of certain material, then the specific procedure should be shown in the monograph of that material. Okay, and then some specific tests can be there, like physicochemical characterization, how to do them, how to perform them. Okay. The There are some specific tests for pharmaceutical water that you should determine the characteristics of this water, for example. Some tests for functionality and safety issues. Okay. Why we do them? Because they affect the product performance at the end. For example, particle size distribution can be determined by these two techniques, analytical sieving and laser diffraction. Why we need to do it? Because it will affect at the end the weight and content uniformity of tablets, for example, if we are doing making tablet formulation. Surface specific area, we do it by gas adsorption. Why? It will affect many characteristics like weight and content uniformity, disintegration and dissolution, and so on. How do we do flowability? We measure the powder flow test. We make powder flow test, and then why we do it? Because it affect also the hardness and compressibility, weight and content uniformity of the tablet, and disintegration, and will affect also that's these two characteristics mainly. Okay, the weight and content uniformity and disintegration and dissolution, and so on. You can go through this table. You have the list of physical properties of particulate solids, of particles, and how do they affect the characteristics of the dosage form, and what are the tests used to identify them. Okay, the last issue we talk about is, can compendial monographs provide test for functionality? No, not directly and not all the time. Okay, the current regulatory environment and the paradigm of quality by design go beyond simply identifying excipient function and emphasizes performance through the identification, evaluation, and control of critical material attributes that assure consistent performance throughout a product life cycle. What is the meaning of this sentence? As I explained to you before, in example of sustained release tablet, pharmacopoeia will just tell you tests to do for release properties and for characterization, just to how to know the viscosity of this one, measure it, and then it should be in a certain range, but there will be variability. How variation within this range will affect my product performance, this you cannot know from pharmacopoeia. How to know it? You have to do it through quality by design experiments. Okay, quality by design experiments. During development phase, you have to vary the attribute of the material, critical material attributes. For example, the viscosity, as an example of hyopromellose performance that I give to you. And then you vary this attribute and you study its effect on the drug product performance, which in my example I explained to you was the drug release rate. And by this quality by design, I vary within a range. I saw and then I study the effect on the critical material attribute, the excipient characteristic here, which is viscosity in my example, on the performance of the product. Okay, and then based on this, I can know really what is the relation between, or the effect of the attribute on the functionality of this material, in my example drug retaining polymer. Okay, so specific test or specification may not be able to determine all the performance related quality material attributes of excipient. So you have a chapter you can read, have a more reading about it in USP 1059. We have a copy of the pharmacopoeia, and you may find some copies online for these monographs, separate monographs. It provides an overview of typical material attributes for many functional categories, along with additional tests. So different functional categories. What are the functions? I have it, but no time to show it to you. I will attach to Caleb. And what tests you should perform to verify the functionality or the excipient performance. Okay. So for you have to choose appropriate test and specification that are necessary to ensure consistent and reliable excipient performance. And this requires understanding of the formulation and manufacturing process, understanding the dosage for performance requirement, and the physical and chemical properties of each ingredient in the dosage form. So before my example, again, I want to make or design a sustained release tablet. My drug is lipid soluble drug, for example, so I know that using a hydrophilic polymer is the most suitable technique. So I have a list of hydrophilic polymers. Okay, I choose several of them. I can study them and the effect on my performance, my target performance, drug release rate. I have to study the compatibility. I study many issues, and then based on that, the compatible one that give me the required performance, I can use it in my formulation. I design experimental design to study the effect of the most important attribute in my knowledge. Viscosity of this drug is the most viscosity of this excipient is the most important attribute that affect the release rate of the drug. So that's why I have to study the viscosity, and the most important characteristic that I have to test when I receive a new batch is the viscosity, and based on this I decide how much of the polymer I should use in my tablet formulation. You have to adjust. Here the last slide is an example of the effect on functionality of effect of critical material attribute on functionality of the excipient. Magnesium stearate as an example, we mentioned before that we use as lubricant. We found that lubrication properties of magnesium stearate is affected by surface area. Surface area is affected by particle size, right? And this can impact the product properties. Okay, what property of the product? Tensile strength of the tablet. How much hard is the tablet? Tensile strength of the tablet is the hardness of the tablet. Okay, magnesium stearate from different suppliers, when I buy from different companies, it has different particle size, and subsequently it has different surface area. Okay, when I look at the pharmacopoeia, in the monograph of magnesium stearate, there is no specification about particle size or surface area. However, from my research, I found that it is an important attribute, and I have to study it for magnesium stearate. Okay, so do I need to study the surface area of all the lubricants? If I use different lubricant, do I need to characterize it in terms of surface area? No, because it doesn't apply to other lubricants. This problem only, or this characteristic only in magnesium stearate. So here, because this affects the strength of my tablet, when the surface area increase, I can see here that the means particle size increase. I can see that the strength of the tablet is decreased. Okay, so if I want a hard enough tablets, I want to use a smaller surface area or larger particle size, in this case larger particle size magnesium stearate. Okay, the last thing, is about impurities. There are many examples that impurities of the excipients may interact with the active ingredient and cause its destruction. There may be some peroxides, aldehydes, okay, interact with the drug directly and cause drug stability issues. It causes degradation of the product or chemical reaction with the product. And here are examples of different drugs and impurities in some excipients and that they interact. Examples of drug incompatibility with excipient impurities. So lactose here, when used with a drug and the drug concentration is about 10% weight by weight, it has some reducing sugar as impurity and they interact with fluoxetine HCl, for example. Raloxifene, it will interact with peroxides that are in povidone and copovidone when the drug loading is about 12.5% and so on. So these are examples of interacting impurities. That's why there should be among the characterization of impurities also I should determine the impurity level, and it should not exceed the dangerous levels that may cause destabilization of the product and the API in the product. Okay, that's the last thing in today lecture. Any questions? Any questions? Okay, then see you in the next lecture, inshallah.